Hello, my name is Cody Kuzno. I am a third-year doctoral student at the University of Michigan School of Public Health in the Nutritional Sciences Department, studying how genetic variation affects ketogenic diet outcomes and ketone body metabolism. During periods of carbohydrate restriction, skeletal muscle and the brain rely on free fatty acids and ketone bodies for fuel, likely increasing the rate of disposal of ketone bodies. Data in the literature suggests that a ketogenic diet, which is low in carbohydrates and higher in fat, and results in the production of ketone bodies, would upregulate mTORC1 in the muscle. Therefore, we hypothesize that this upregulation of mTORC1 enhances ketone body disposal through some underlying mechanism. We first developed a ketogenic diet and a protein-matched control diet. To test if the ketogenic diet was sufficient to produce a state of ketogenesis, a strain of mice, known as AJ, were fed with either diet for 3 weeks. As shown in the graph on figure 1a, mice fed the ketogenic diet exhibited increased serum ketones for both male and female mice compared to control diet-fed mice. Also shown in figure 1b are the body weights of these mice. Mice fed the ketogenic diet also presented with lower body weights compared to mice fed the control diet. We then developed a ketone tolerance test where the AJ mice were fed either a ketogenic diet or control diet for 3 weeks and were then injected intraperitoneally with 1 milligram of beta-hydroxybutyrate per kilogram of lean mass per mouse. Beta-hydroxybutyrate is the main ketone body formed under ketogenic conditions. As demonstrated here in figure 2, ketone tolerance appears to be improved with ketogenic feeding. Next, to test if muscle mTORC1 activation is sufficient for driving the enhanced ketone body disposal seen, muscle-specific TSC1 knockout mice were generated in lab. The ablation of TSC1 in the muscle of these mice should result in the upregulation of mTORC1 activity, shown in figure 1a. This upregulation was confirmed by Western blot analysis, shown in figure 3b, of various muscles where we see the knockout, which is squared off in red on the figure, leading to increased phospho S6 over S6, indicating upregulated mTORC1. Quantification of the Western blot analysis, shown in figure 3c, confirms the increased phospho S6 over total S6 in knockout mice, again squared off in red, compared to wild-type mice. Now, to test whether upregulated mTORC1 had any effect on ketone turnover, we again performed a ketone tolerance test on knockout mice and wild-type MTSC mice, where again 1 milligram of beta-hydroxybutyrate was injected intraperitoneally per kilogram of lean mass per mouse. Blood ketones were checked every 15 minutes for 90 minutes, and figure 4 demonstrates the results from that experiment, where in the knockout condition, mice exhibited increased ketone body disposal compared to their wild-type counterparts. Last, we performed mRNA analysis in the muscle of these MTSC mice. Figure 5b shows the mRNA analysis of quadriceps muscle, and this analysis demonstrated elevated expression of ketone transporter MCT1, encoded by SLC16A1, D-beta-hydroxybutyrate dehydrogenase, encoded by BDH1, which catalyzes the interconversion of acetoacetate and beta-hydroxybutyrate, and the rate-limiting step of beta-hydroxybutyrate into acetyl-CoA, SCOT, which is encoded by OXCT1. Therefore, we conclude that TSC1 ablation in the muscle leads to increased mTORC1 activation and results in improved ketone body disposal. We predict that this increased ketone oxidation will lead to reduced hepatic triglycerides and improved insulin sensitivity that could result in improved body composition of these animals on a ketogenic diet. Thank you.